Seyfert-Margolis here. She is currently the founder CEO of Immunotherapy, which is a mobile application platform technology for family-based co-management of health. And then prior to this, Vicki was a senior advisor for science innovation policy in the Office of the Commissioner at the FDA. And her focus there was the development and execution of a agency-wide overarching strategic plan for regulatory science. Prior to that, she was CSO at Immune Tolerance Network, as well as serving as director of the Office of Innovative Scientific Research Technologies at the National Institute of Allergy and Infectious Diseases. And education-wise, Vicki completed her PhD in immunology at the University of Pennsylvania School of Medicine and her postdoctoral fellowship work at Harvard University and NCI. So we're really pleased to have Vicki here, wrapping up the individual talks. With that, take it away.
Okay. So, Mary Ellen twisted my arm to come and give this talk. When I was the CSO of the Immune Tolerance Network, we were among, I would say, one of the rare organizations that was involved in developing leading-edge assays and bringing them to GLP, CLIA, and in some cases, FDA compliance. And at the time, starting back in 2000, what we were engaged in was development of leading-edge technologies, assay technologies, that would help us understand correlates between therapeutic interventions, namely monoclonal antibodies, cell-based therapies, and peptides for treating autoimmune disease, asthma, allergy, and solid organ transplantation. So it was a broad mandate. And our goal was to develop correlates that would hopefully turn into predictive markers. And back in 2000, I'm dating myself, I think it's really important to understand that the landscape in the pharmaceutical industry and in the clinical research industry was one that was very focused on an endpoint. And when we started, most of the companies, and we worked with almost all of them, major pharma and many of the more larger biotechs, all of them were interested in biomarkers, not so much. They really were like, look, we're here to get our endpoint, prove our efficacy, and sell our drugs, and we're not sure we want to know more about subpopulations that actually respond to therapy or not. I can't help but think about how much the world has changed, and I'm going to talk a little bit about that with respect to the complete integration now of assays into clinical trials and into clinical research. So much so that companion diagnostics is really part and parcel to almost all drug development now. My first project when I came to the FDA was to get the companion diagnostic guidance, draft guidance, which had been sitting sort of in the bowels of the agency for 5 years before I arrived, and get it out the door so that we could provide to the community the science and the regulatory policy considerations in building a companion diagnostic drug development program. So when Mary Ellen came to me and said, can you talk about regulatory and bioassays? I said, okay. It is a big area, and I'm going to try to talk about two big topics that I think are on front and center on the FDA's mind right now, certainly when I was there, and I think still to this day. One of them is around companion and complementary diagnostics and assays and how they're being used, and the other is around potency assays for advanced therapies, and namely really CAR-T and oncology driving both of these areas in a major way. So I think it is fair to say that oncology drug development is really on the leading edge of clinical trials and assay development right now with respect to how assays are being integrated into care delivery and how assays are really now integrated into drug development. I'm sure you spent the day talking about many nuances of assays and controls and qualities and many of the in-depth things that I may or may not understand. I should say I would understand them from my days, but may not be completely current on. I will put in one, I guess, plug, shameless plug for what I'm doing now. When Mary Ellen described a mobile care management, really what I'm doing is using digital technology layers to support real-world evidence studies. I have a paper, a commentary that just came out in Nature Biotechnology this month, which explains what we do. And the reason I'm talking about it is I think at some point the bioassay community is going to be talking about digital assays, because the data that we're thinking about in understanding and making measurements of the biology people is going to be merged with data that's coming in through sensors and mobile technologies. And I think that that day is arriving. So I like to think of the biomarkers world as biology biomarkers and digital biomarkers. And so I'm currently involved in the digital biomarkers business, which has many parallels, quite honestly, to biomarkers and bioassays. All right, with that, I tried to divide, for the purposes of this talk, the universe into two key areas. And again, I went through sort of the latest, greatest coming out of the EMA, as well as what I saw was the latest, greatest coming out of the FDA. And I think we are still struggling as regulators and as developers in both these areas. The first area is where the assay is a diagnostic test to direct therapeutic treatment. We all know that analytical validity, and I'm sure you have spent a lot of time talking about analytic validity today, is critically important. But I can tell you that every time I was at CDRH and every time we had a discussion about assays, whether they were direct to consumer, whether they were LDTs, whether they were IVDs, whatever they were, clinical utility was always the hardest thing to really demonstrate and to prove. Where and how is this assay really getting us to some clinical decision that's being made? And what is the risk of that assay and the data that's coming from that assay to the clinical decision that's being made? Now, that may not be in your universe or your world if you're doing research or you're doing high throughput screening or you're doing assays for preclinical work. But at the back of everyone's mind, I really think we need to be thinking about this through the entire product development life cycle and continuum, which is whatever we're doing in research, whatever measures we're making, at the end of the day, need to translate into something that matters to patients and to their care. And this is really not that easy. So predicting the performance of a therapy is tough. And that means really digging in and trying to figure out what are we measuring, how precise are our measures, and what does that mean when we have a patient in front of us? I would say that I can't tell you how many assays I did on clinical research samples comparing normal controls in patient with disease or patient with disease pre-therapy and patients with disease post-therapy. And every single time the data was like this. There was some signal that the control group had and some signal that the affected group had, and there was always overlap. So that for whatever we were measuring, there was always this concern that, again, when you have an individual person in front of you with a diagnosis, where are they on this continuum? And how do we make a decision based on the data? So I think that's really important. It's also really important to note that right now, and again, oncology leading the way, clinical trial design is in large part being driven by drug diagnostic combinations. And so these assays are becoming more and more central to everyday therapy development and everyday use in the clinical stewardship of patients. The second case that I'm going to spend a little time on that is the assessment for a GMP or performance of the complex therapy itself. It was interesting because, you know, when I first came into the agency, a lot of the innovative business was at CDER, and there was a lot of discussion about where biosimilars would reside, because it was previously in CBER and then moved to CDER. And certainly there's a lot of interesting and important work around assays and potency for biosimilars that's being dissected and being developed right now. But clearly the leading edge, and I think of complexity, resides in cell-based therapies. And the struggles that CBER has with coming up with accurate assays to reflect what is actually being delivered in a cell-based therapy and what will happen when we deliver these cell-based therapies into people. So I'm going to talk a little bit about that. And with the two recent approvals of CAR-T, one for Kite and one for Novartis, you know, the future is here, and we really have to think about that. I want to make sure we distinguish that from minimally manipulated therapies, because in the world of homologous cell-based therapies, where we're taking tissue or cells out of people and doing something to them, putting back in, there is a whole universe out there, a lot of it in cosmetics, where this is going on that is not under regulation right now in the strictest sense, because they're considered minimally manipulated and clinical. So we won't be discussing some of those things. I'm going to stick to cell-based therapies where there's manipulation involved, and CAR-T, of course, is a prime example. I also came upon a paper that I thought was very interesting, and I had some discussions when I was at the agency about some of these novel therapies, which are antibody-drug conjugates, you know, basically the targeted toxin-type approach for cancer, and bispecific antibodies, where there's two different FC components on a single antibody chain that have been engineered, and the challenges and difficulties in setting up assays to understand the potency and the dosing around the use of those therapies. And I'm not going to spend a lot of time on it, except to say this is, again, another area that's really hard, really challenging. When I was at ITN, we were simply trying to do a dose titration for a monoclonal antibody therapy, anti-CD3. And we spent months and months and years to make sure that we had the best controlled flow cytometry-based assay, competition assay, to really try to understand how we could build models that would predict dosing and efficacy in vivo. This is really hard stuff. And I think these novel therapies are going to make it even tougher. So I hope that this is of interest to you. Maybe it's outside of what you do, but hopefully I'll be entertaining enough, or you can go to sleep because it's the end of the day, or you can go get an earlier flight. Okay, so companion diagnostic assays. One of the interesting things to me is companion diagnostic assays are really about selecting people that are going to respond to therapies. And I don't know if any of you have been following the literature, but if you look in cancer, again, there are two big protocols out there right now. One's in breast cancer at UCSF, and one's called LungMap, that's across the country. And the thing that's so novel about these are a new trial design called basket trials, basically that are predicated on diagnosing people or running people through a series of assays, primarily genetic assays, looking at sequence information, sequence variants, and then shunting them into a treatment modality. So there's 5 different arms and a Bayesian statistical approach to the breast cancer study. And LungMap, I think, has 5 or 6 different arms and then several sub-arms within it. And I think the critical thing about this is that companion diagnostics are being used and are FDA guided in these 2 cases for doing novel study design, where the assay is central to what is going to be the intervention of choice in these clinical trials, and hopefully will become the care model upon which decisions will be made and built in breast cancer and in lung cancer. So we know companion diagnostics are supposed to identify patients who are likely to benefit from a particular therapeutic product. And I like to say that in oncology, they are leading the way because we have a nice kind of uniform tumor which we can sample, as opposed to a disease like diabetes, where what are we sampling? So we really are predicating a lot of companion diagnostics on the biology of the tumor to date, and not really the biology of the patient. And I think that's really important to keep in the back of our minds, that there's going to be underlying biology of the patient that also is critically important in cancer. But we're starting with the tumor, and we're starting with a very clear mechanism of action of a drug that can be distinctly and definitively linked to a bioassay, in this case, mainly gene sequence information. However, there is this other thing that's emerged with respect to identifying patients who are likely to benefit, which is called a complementary diagnostic. And this is really being driven, again, in oncology by the use of immuno-oncology therapies that target the PD-1 pathway. So Keytruda and Opdivo are 2 drugs that are on the market right now with multiple indications. But the key question that confronts us about the use of those drugs are, does the tumor express PD-1L? Is the drug going to work? And if the tumor does express it, what level would correlate with the response to those antibody therapies. And if the tumor doesn't express it, that we can measure, is it possible that the patient still will respond to the therapy? And so right now in the clinic, the PD-1 assays are called complementary diagnostics, and they're being used to try to guide whether or not someone's going to respond to Keytruda or Opdivo. But they are not well enough defined, and the studies are not known yet in terms of the clinic whether or not it's an absolute predictor. So you can see at the bottom, you may have somebody who doesn't have measurable levels of the PD-1L, but they may still respond to therapy. It could be because it's below our limits of detection, or it could be that there's an independent pathway which the antibody is triggering, and we don't know yet. So we're using this assay in the clinic, but we're not using it definitively as we are with many of the other genetic assays. These diagnostic tools are also used to monitor adverse events or to predict adverse events. Many of the cytochrome P450 mutations have been instrumental in sorting out dosing differences or toxicities for people, and then of course monitoring responses to treatments with a particular therapeutic product for the purpose of adjusting it and really seeing whether or not the product is working. So when the FDA first issued the in vitro companion diagnostic guidance, it defined it as an IVD, and I want to be clear, this was initially defined as an IVD, so to get your product on the label, it was part and parcel to the regulatory approval that provided information essential for the safe and effective use of the corresponding therapeutic product. It described what a companion diagnostic is. It clarified that they expected contemporaneous regulatory approvals, meaning you had to set up your pathway, and this is not easy for your drug development and your diagnostic development to coincide. They're very different paths, and they need to be set up to coincide. That's not easy. And described some of the regulatory requirements. What they didn't provide at the time was how to do it. They said basically, here it is. We'll know it when we see it, but we're not going to give you too much guidance on that. So more recently, they did issue a second guidance or draft guidance on how to do it. And I think there's some important elements in here that, again, I just want to underline the point that everything we're doing in the world of assays could lead to something or could lead to a product that's going to actually be used in the clinical trials. So we need to be thinking hard about how it might be used. So the idea here is that the therapeutic product development and the assay is happening jointly in preclinical. This is not something that's just happening in clinical because, again, the assay is going to be connected to the MOA of the drug. And that there needs to be check-ins, essentially, between preclinical and early stage development, and then, of course, after the early stage trials and the pivotal trial that's being done. So whether the studies are initiated at the outset of development or at a later point, most of these are going to be initiated at the earlier points. They have to be conducted in a way that obtains contemporaneous marketing. So again, you can see that the idea here is you've got to be able to time your development of your assay performance so that it coincides with your drug. They also laid out some biomarker-based clinical trial designs, and I think this is really interesting because if you follow a lot of what's been going on with oncology, it's been more like B, where there's a marker that's linked to the mechanism of action of a drug, and the person is tested for that, and if they're positive, then they're randomized. If they're negative, they're left out of the study. And what this leaves the agency and with all of us in a bit of a conundrum about is, what is the MPV? Are there potentially responders that exist within the negative population? I'm sorry, the PPV and NPV. Are there potentially responders that exist that are never being tested for these drugs in clinical trials because the trials are predicated, or entry into the trials are predicated upon being positive for the marker? So I think it's really important to think through, again, how definitive do we think these assays are, and are we, using the best trial designs to be able to test the clinical utility of these markers. The other alternative, of course, is to test for the marker and enroll either people who are positive for the marker into the treatment or who are negative for the marker into the treatment. I can tell you that there are a lot of issues ethically about this with respect to whether or not you would enroll people into a treatment trial with a therapy that carries risks when you firmly believe that if they're marker negative, there's no chance of benefit. So this really leaves us in a conundrum about whether or not we should be testing drugs that we think are clearly linked to a diagnostic test in people who do not possess the marker and therefore should not, by all of our biology knowledge, respond to therapy. So in terms of the co-development process, some of the critical points along the way where CDRH and the IVD component is lining up with the study are shown here, and this is straight out of the FDA guidance, meaning that the IVD is going to be central and part and parcel to the phase 1, 2, and 3 studies, and that as the drug moves along in its product development, so must the assay to move from an analytically validated test all the way to the final analytical performance characteristics, which are determined and are definitive as diagnostics for that person and the decision being made about putting that person on the therapy as someone who will likely respond to the drug. It should be noted that the label itself will reference the test and will reference the IVD that went in for approval. Here's where things start to get a little bit interesting, however. There are 18 drugs that have been approved with a companion diagnostic. 3 of them utilize next-generation sequencing testing. There are many IVDs that are approved with these drugs. However, there are abundant numbers of LDTs that are also out there. I assume all of you know the difference between IVDs and LDTs. Is that correct? No. Okay. So the IVDs are tests that are regulated by the FDA, and IVDs can be used in a kit or can be distributed across state lines because they are regulated by the FDA. Laboratory developed tests, or LDTs, are tests that are performed in hospital laboratories and are regulated by CLIA, which is the Clinical Pathology Laboratory Organization, and are governed and overseen by states, so that each state has different requirements. Many of them are similar, although New York State and California have additional requirements on top of sort of typical CLIA that must be adhered to. The testing of those assays that are done in CLIA labs are outside of the jurisdiction of the FDA and have to just simply show reference ranges and analytical validity for those tests. The challenge is, and Mary Ellen and I have spent a lot of time talking about this, is if you have an IVD-approved test that went in for the diagnostic to determine whether or not you do a drug, and it's a particular sequencing technology or it's a particular immunohistochemistry technology with a particular variant that you're looking at, and then you wind up going to Memorial Sloan Kettering, you will find that Memorial Sloan Kettering is not using the IVD test. Rather, they are using their own panel and their own LDT of sequence variants that they've developed in-house and are regulated under New York State law. That test may or may not conform to the specifications of the IVD that was approved. So the question is, for the purposes of the patient, are we serving the patient population well by having slews and slews of different LDTs that exist out in the community? This is a big issue. It's a political issue. It is an issue that's been discussed on the Hill countless times. When I was at the agency, there was a discussion about enforcement discretion. Enforcement discretion means the FDA decided not to try to regulate LDTs, but they reserved the right to. They were getting ready to regulate the some of the LDTs that were considered high risk. This would be ones like, hey, I've got lung cancer. Should I get Opdiva? Do I have the gene? And am I being tested with the right test? Is the test at the community hospital that they're doing locally going to be as good as the test that was developed with the drug? And when the change of the administration came in, it was decided that that would not be enforced, that LDTs would not be regulated. So I think one of the things that I want to sort of drive home about this is not that I'm opposed to LDTs or that I think they should be regulated, but it's really important for patients and really important for practice that we start to think about standardization and references, because right now it is Wild West out there. And I'm not saying the FDA needs to do it, but I am saying that there is a lot of room and a lot of need to think about how, when we take these advanced technologies, we move them through a regulatory path. There's a ton of copycat assays that immediately are built and made and will be done as LDTs. It also takes a lot of the economic incentive, by the way, out of developing assays for people to go through the FDA because they don't get a nice goodie that you get with a drug, which is basically exclusivity for 7 to 12 years. You don't get that. So the big question is, how good are these assays performing, and should everybody have their own cottage industry for these really complex assays and the decisions that are being made from them? So Foundation Medicine is a panel. As I said, Memorial Sloan Kettering has a panel. Many of the cancer centers have panels. Foundation Medicine is one of the panels that's often ordered at different clinics, and they just recently have gone in for an FDA approval. They're right here in Boston. They decided to take that path. Similarly, for the complementary diagnostics, and I want to, just bring home the point, these are being used for people who have cancer, and I've, by the way, unfortunately had the opportunity to be sort of the shepherd of several people, friends of mine with serious cancer, right now 17-year-old with osteosarcoma, who's one of my colleagues' nephew, and just watching the confusion of these tests on a cancer patient and how they're being used in clinical decisions that are being made is just really frightening. So we need to do a better job. Right now for PD-1s, for the PD-L1s, there are 4 approved diagnostic tools with 4 different antibodies on 2 different immunohistochemistry platforms and 4 scoring systems that have not yet been harmonized. This is the best it gets. So, again, the question is, who's going to step in? What's the responsibility of the community? So some of the big policy challenges for these companion assays are analytical performance characteristics and standards. What are they? Where are they? Who's responsible for them? There are none. Analytical validation across labs, not happening. Real-world evidence about test performance. Well, as it turns out, in the study that I show on the right, it turns out that the companion diagnostic tests for these particular genes did quite well in terms of the LDTs and the IVDs performing pretty equivalently. But one of the things they discovered was that each lab that's performing these assays does their own little tweaks. And even on the IVDs, which are sort of kitized, they're still doing their own little tweaks, which may be around how they collect the sample, how they run the test, et cetera. So understanding what's actually going on and what's affecting test performance is critically important. Changes made to the assays when they're being implemented, as I just mentioned, and then of course changes made due to advances in the technology. How do we reference back? How do we know to keep current and make sure that we're providing the best assays for our patients who are in the clinic? So are there any questions on this part? Because I'm going to switch gears to the cell-based assays. Or should I wait till the end? Yeah?
So how about that if certain cancers have more mutations, and then if they are doing assay a particular X test, but that particular patient is not having that X test, but there is a Y test that has to be, but the cancer, that malignancy or the cancer that have the patient is the same, but the mutation was different.
Right.
So, but they can claim that when we tested with whatever test we have, it's shown a negative, but there is a subtest that
right.
need to be addressed.
Right. So this is part of the challenge because a lot of the laboratory developed tests that are being done on a specific, what I'd call refined or honed panel that is a subset of the known mutations that exist that are particularly important or have been defined as important in sort of the research context for cancer and for therapeutic decision making. So companies like N of 1 and Foundation do a panel that's pretty complete. The problem with that is that the test costs about $4,000. It's generally not covered on many insurance plans. So you'll get a more comprehensive test out of Foundation, but the question is whether or not you'll even get to the test. Who will order it and who will pay for it? When you do the test locally, you'll probably get a subset of what I would call the most popular candidates, and it may be done with a methodology that's not as sensitive or specific as some of these other tests like Foundation. So you may miss other mutations that are important or could be important in the therapeutic decision making. And of course, this means that we have sort of inequality in the care that's being delivered. And I think it's just a really important point of how we've seen assays mature into these very powerful clinical tools, but we're not seeing them used equivalently and probably as best as they could be based on all this leading edge science. Okay, so I'm going to move on to another complicated area. So bioassays for cell-based immunotherapy products. So this is a slide I thought was interesting that I pulled from the FDA, the MOA in the human body, a learning process and product life cycle. So we know that we can measure, or we can attempt to measure things that are going on in vivo, meaning in vivo in people, using a variety of strategies, enzyme assays, cell-based assays, or relevant disease models. But we also know that they generally don't capture all the functional attributes of a product, like oscillation, ADCC, CDC, clearance, all the different things that are happening in the body. But we know they're an important point right now in trying to assess the potency of these complex products that we're putting into people. So these assays can be done in vitro or in animal models. More commonly, they're being done in vitro. They're based on a defined biological effect that's as close as possible to what we believe the mechanism of action is. And again, I'm going to talk about cell-based therapies where we think we know, but I'm sure there's a lot of other stuff going on that we don't know. And the validation of these assays really needs special consideration because of the complexity of the product. And again, I think this is regulation in flux. If you look at the evolution of bioactivity potency regulations, you can see the continuum over time. And really the interesting thing is that not since 2011 has there really been much action in terms of a formal draft guidance coming out of the FDA. The line on the top are new complex antibody therapies that have been developed since this guidance has been released. And then recently, the EMA released a position paper on potency of assays, and I have those highlighted on the right. The guidance for industry, potency tests for cellular and gene products that the FDA has released, the European Medicines Agency, the EMA's guidelines in potency testing. I would recommend reading this. This is a short document that's actually quite thoughtful. It's focused on immuno-oncology because, again, that's where the pain is right now for these cell-based therapies. But I think there's a lot of important considerations in there. And then this is the ODAC, which is the Oncology Disease Advisory Committee filing for the Novartis product that was approved, namely Chimera. And I just wanted to show that CAR T cells, which are now on the market and cost a lot of money, so this is listing for about $350,000 to $400,000 per infusion for this therapy, have a very complex manufacturing process, which is shown here on the right, involving removal of cells by leukapheresis, T cell activation, introduction of a synthetic receptor, expansion of the cells, and then reinfusion with a combination of chemotherapy. And all along the way, assays need to be performed to show that you have potency, purity, lack of contamination, and that the manipulations that you're performing on the cell are working, and that you have, at the end of the day, bioactivity. So the activity that underlies this involves the use of a transfected cell with a receptor that targets CD19 and basically kills B-cell lymphomas. So this product has been approved. What I wanted to show you that I think is interesting is this is the actual assay data from the filing that Novartis put in as part of their approval for the CAR T product, which was approved. And you'll see the data is pretty all over the place. But this is what it looks like. So these are the results of the percent of CAR T positive cells which were transfected, and the levels, the percent of CAR T positive cells that are in the pool in their manufacturing process shown on the left. And on the right is their bioactivity assay, which for them they believe was gamma interferon production per transduced cell. This was produced during culture with the CD19 expressing cells, which I told you was the target. Quantified as a measure of potency for the efficacy of the drug, or I should say drug in this case, cell-based therapy to be infused. So I wanted to just point out how challenging this is, and there are cutoffs that they try to establish based on having a certain proportion of these cells which express a certain level of the receptor and which produce a certain amount of interferon gamma. Now, this is by no means an exact science, and we really don't know yet, except for the limited clinical trials, where on this continuum really leads to optimal efficacy. But this is an example of where we are right now in the assessment of cell-based therapies which are approved and going into human beings for treatment. So a couple of things to talk about in closing is today's innovative bioassays present many challenges on the policy side and many opportunities that are not just the responsibility of regulators, but I think the responsibility of the field, but will fall in the hands of the regulators ultimately. The FDA and the EMA need help from the community to describe materials that are acceptable for validation given the limitations of clinical samples. So it's not like, you can get a breast cancer sample that's got a certain mutation in it and there's enough tissue to distribute across all the labs in the United States. There's just not. So we need to really work to identify reference standards and to get material together, whether it's cell lines or whether we have pooled tissue or whatever it may be, to allow us to do comparisons and proficiency testing at these various locations where assays are being performed. The National Institute of Standards and Technology should be encouraged to develop standard reference materials with the community that's made available to sponsors and labs. Right now, all of us are working with our own standards and our own reference material. Multi-stakeholder groups like this one should identify high-quality reference materials for analytical performance. I guess I said it before, I'll say it again. The FDA doesn't have a repository of cells, they don't have a repository of tissues, and they don't have a repository of knowledge that exceeds that outside in the field where people are working on assays. They need help, they need input. And the community needs to think about this and come up with some suggestions. Real-world evidence needs to be gathered about test performance and patient outcomes. With all of these laboratory-developed tests that are out in the market right now, we need to know how comparable they are. All of us are going to experience either someone we know or, unfortunately, ourselves that has cancer. Now, all of us will be impacted by this. And as I said, I've done this a couple of times. It is not easy to say, gee, you had the test at MSK. Should we get the test at Foundation? Should we get the test at the local hospital? Who has the data? Where'd the data go? Does that mean you should be put on this? Are you going to be put on this before you've had chemotherapy? Are you going to be put on it after you've had chemotherapy? It is really hard. And if we don't have the utmost confidence in all the tests out there, then how much confidence can we have in the decisions that are being made? Proficiency testing data needs to be shared. There needs to be sharing among the community about what you know and how you're doing it and standardization of those methods. And of course, we need to be providing more information to physicians and patients. How will the physician know which assay to use? How do they know whether the assay being done in their hospital is as good as another one, or whether the assay that they bought, from Roche is the best assay at the time? So we really need to get more information out to physicians and patients, and I know they're confused. And this line actually came to me from a patient advocacy organization. They are very interested in how do we get more information out into the community for using assays. So I'd like to close with this idea of the cycle of bioassay virtue of value. We need to establish assays that are fit for purpose. That means threading all the way through through product development. The assay that you used in research and preclinical may very well have important implications in the clinic once the drug or the thing that you're trying to measure is out there being utilized. We need to generate metrics and methods for validation. This needs to be done among the community, between the community, sharing of information, sharing of data, and of course sharing of reagents. Use and share reference standards across labs and share methods. I can't speak enough, as Mary Ellen knows, I've been an evangelist for this for a long time. Reference standards, reference standards, reference standards. The FDA wants them, they need them, they don't have them, they don't know what's out there and what should be used. So the community needs to really get active on this. And then bringing validated assays to the clinic with information, good information to support the use of the assay. If you're going to, take an assay and use it in 5 different places, make sure the 5 different places are doing it the same way. And that brings me to this idea of analytical validity, clinical utility, and our ethical and legal and social implications and responsibility that we establish and we use these technologies in the context of use to the best of our ability and the best circumstances, because people's life-saving decisions are being made on these assays that we have out in the, in broader use right now. So that may not be what you guys were talking about all day, but I thought it was something a little bit bigger picture, and I'm happy to take questions about anything that I can answer with respect to FDA time, and I won't stand between you and your drinks too long. Oh, right, we have a panel. Thanks.